Hello everyone. Today in this video we will learn regarding oxidative phosphorylation. So this is the second step of ATP synthesis. The earlier step includes electron transport chain. To learn regarding the electron transport chain, please do visit my previous video regarding electron transport chain, wherein the detailed steps of the electron transport chain have been defined and have been explained. So now this oxidative phosphorylation helps in synthesis of ATP. So this oxidative phosphorylation we will learn the different mechanism, how it is being carried out, and how the rotary model of ATP synthesis is being carried out. And also we will learn how the oxidative phosphorylation is being inhibited and what is the significance of these inhibitors. So let us start. So the electron transport chain through the ETC is linked with the release of free energy. As we have already learned that during the transfer of electrons from one complex to the other complex in the electron transport chain, there is also liberation of the energy. So that energy is utilized for pumping of protons from the mitochondrial matrix into the intermembrane space. Now, whatever the intermembrane space protons are present, the concentration of those protons goes on increases as the electron transport chain proceeds. So at the final step, that is after completion of the fourth complex reaction, and when the protons are highly accumulated in the intermembrane space, these protons try to come back again into the mitochondrial matrix. But the mitochondrial matrix, that is inner membrane, is highly impermeable to protons. So what happens? These protons try to travel through ATP synthase. This ATP synthase is the fifth complex of electron transport chain. So when they travel from this ATP synthase, there is change in the conformation of this ATP synthase enzyme, and energy is also being released during this conformational change. So this energy is utilized for conversion of ADP to phosphate, resulting in the synthesis of ATP. So this is how exactly the oxidative phosphorylation takes place. So the complex 5 of the inner mitochondrial membrane is the site of oxidative phosphorylation. So here important point to remember is PO ratio. What is this PO ratio? So PO ratio represents the number of ATP molecules synthesized. Number of ATP molecules synthesized per inorganic phosphate and oxygen consumed. Means for each atom of oxygen and for each atom of phosphate, how many ATPs are synthesized is known as PO ratio, that is phosphate and oxygen ratio. So as we all know that NADH, during oxidation of NADH through ETC, 3 molecules of ATPs are produced. So this means that the PO ratio of NADH is 3. The PO ratio of NADH is 3, that is it utilizes 3 oxygen atoms as well as 3 phosphate atoms that are converted into ATP, that is 3 molecules of ATP in presence of 3 molecules of ADP as well. Similarly, for FADH2 there is 2 ATP synthesis and the PO ratio of FADH2 is 2, means 2 oxygen atoms and 2 phosphate atoms are being consumed during the oxidation of FADH2, that results in synthesis of 2 molecules of ATP. So this is the significance of PO ratio that defines how many number of molecules of ATP can be synthesized by oxidation of these coenzymes. So coming to the mechanism of oxidative phosphorylation. So during this mechanism of oxidative phosphorylation, different hypotheses have been proposed. So these hypotheses are, one is chemical coupling hypothesis and the second one is chemiosmotic hypothesis. So what are these different hypotheses? According to this chemical coupling hypothesis, this was proposed by Edward Selter. You can see Edward Selter in the year 1953. So according to this hypothesis, there is a coupling reaction. You can see the name indicates chemical coupling. So there is a coupling reaction between the phosphate groups and high energy intermediates. Whatever the reactions are being carried out during the oxidation of glucose by glycolysis, PDH complex or Krebs cycle, and finally to the ETC wherein the reduced coenzymes are oxidized. So all these reactions are facilitated by synthesis of intermediate compounds. So some of the high energy intermediates are also produced. Like you can remember in the case of glycolysis, the last step, that is phosphoenol pyruvate to pyruvate. So as we have learned in the high energy compounds in bioenergetics topic, we had learned that high energy compound phosphoenol pyruvate is also one of the high energy compound. So these high energy, according to this chemical coupling hypothesis, Edward Selter assumed that these high energy intermediates are phosphorylated. These are first phosphorylated. After the phosphorylation of whatever the energy is being utilized for phosphorylation of such high energy intermediates, that energy is only responsible for synthesis of ATP. So while proving this chemical coupling hypothesis, certain disadvantages and certain drawbacks have been occurred. So because of this chemical coupling hypothesis has been removed. So the new hypothesis, that is chemiosmotic hypothesis, this was proposed by Peter Michel in 1961. So this hypothesis assumes that whatever the protons are pumped into the intermembrane space and whatever the energy is being produced during the electron transfer from one complex to the other, these both, that is the protons as well as the electron energy, these both are coupled, and this leads to the synthesis of ATP via ATP synthase enzyme or via ATP synthase complex. So now this chemiosmotic hypothesis is worldwide accepted, and this was also proved how the protons are responsible for synthesis of ATP from ADP and phosphate. So this chemiosmotic hypothesis has different components. So one of the important component is proton gradient. So what do you mean by proton gradient? So as we know that the inner mitochondrial membrane is impermeable to protons as well as potassium ions, hydroxide ions, including some molecules like ATP, ADP and pyruvate. We had learnt in the last video that the inner mitochondrial membrane is impermeable to ions as well as molecules. So now through the transport of electrons to the ETC, that is coupled with the translocation of protons, we have also learned that during the transfer of electrons from one complex to the other, there is also transfer of protons from mitochondrial matrix to the intermembrane space. So these both reactions are coupled. Now these, after coupling of these reactions, there is a proton increase in the proton gradient in the intermembrane space, resulting in electrochemical or proton gradient. So now this electrochemical or proton gradient tries to pump back the protons from intermembrane space back to the mitochondrial matrix. So whatever energy that is being utilized for pumping back of protons from intermembrane space to mitochondrial matrix is utilized for synthesis of ATP from ADP and phosphate via ATP synthase. So this is how the chemiosmotic hypothesis was proved using proton gradient as a main component. So the other is enzyme system for ATP. As we know that enzyme responsible for ATP synthesis is ATP synthase, that is the complex 5 of the electron transport chain. Now this ATP synthase consists of two functional subunits named as F1 and FO. These subunits, these are also known as F1 particles and FO particles, that is F1 FO particles. So these, here is the structure you can see. This is the ATP synthase, wherein this is the base of the ATP synthase, that is FO. This is embedded in the inner membrane. This is outer membrane, or you can say this is the intermembrane space, and this side is the matrix. So the matrix side consists of F1 particles of ATP synthase, while the inner membrane space is embedded with FO particles. Now these FO particles consist of C units. You can see this in light green colored. These are C units, while the F1 particle consists of 3 different components. One of the component is the gamma component, that is this dark green colored rod shaped, you can see, that is a gamma particle, and it also consists of 6 other particles, that is 3 alpha particles in blue color while 3 beta particles in the pink colored. So F1 component of the ATP synthase consists of 1 gamma rod, 3 alternating alpha and beta components or subunits. So totally 6 subunits alternating with alpha and beta, and 1 gamma subunit on 1 gamma rod that makes a complete F1 particle, while the FO particle consists of C units. So this the hypothesis that was proposed by Michel defines the ATP generation via rotary model of ATP generation. This is a rotary motor model of ATP generation. So in this model was proposed by Paul Boyer. So this Paul Boyer person defined how exactly the ATP generation takes place, and he justified the chemiosmotic hypothesis that was proposed by Peter Michel. So this chemiosmotic hypothesis was justified after the rotary model for ATP generation was proposed by Paul Boyer. So he defined that there occurs a conformational change in the ATP synthesis enzyme. So this conformational change is responsible for synthesis of ATP. The conformational change, whatever takes place during the transfer of protons via ATP synthase, helps in generation of ATP. So this ATP synthase consists of FO and F1 complex. These FO subunits are composed of C subunits, while the F1 particles consist of 3 alpha units, 3 beta units and 1 gamma unit. So this is the structure. Now when protons try to move from the intermembrane space into the matrix, they can only move through ATP synthase because the inner membrane space is impermeable. So during movement through the ATP synthase, there is a conformational change in the beta subunit. Here the point to be noted is only beta subunit conformation changes, while the gamma and alpha subunits conformation remains same, because beta subunits consist of the sites that are responsible for conversion of ADP to ATP. It has certain sites that are responsible for conversion of ADP to ATP. This is the reason why only beta subunits undergo conformational change. So what happens is, when the concentration of hydrogen ions is more in the intermembrane space, they try to move through the ATP synthase, and during this movement, this gamma particle or gamma subunit, this is the rod green colored, it rotates the F1 particle. It rotates the F1 particles. So during this rotation, there is change in the conformation of beta subunit, because ADP and phosphate come and bind to the beta subunits, and there is efflux, or you can say influx, of protons from outer side to the inner side. So there is rotation of this gamma rod. During this rotation, there is synthesis of, or there is a generation of energy. So that energy is captured by beta subunits. Now these beta subunits already they are loaded with ADP and phosphate. So they consume this energy during rotation, and ATP is being synthesized. And ATP is synthesized and is released. And this ATP is stored in our body. So you can see this is finally leads to synthesis of ATP. Now during this conformational change of beta subunits, it exists in different conformations like O, L and T. So what are these three conformations? One is open, loose and tight. Again you can see in this clip that this is a blue colored is an open structure of beta subunit. This is specifically for beta subunit. Don't compare these with this. This is only for the beta subunit, wherein this O is an open conformation of beta subunit, T is a tight conformation of beta subunit, and L is a loose conformation of beta subunit. So what happens is, first ADP and phosphate molecules come and attach to the loose conformation, that is L conformation of beta subunit. So you can see this is a loose conformation of beta subunit. When ADP and phosphate binds, the conformation shrinks and it turns to T conformation, that is tight. So during this tight conformation, the energy is utilized because of transfer of protons, and the ADP and phosphate molecules combine, leading to synthesis of ATP. Now this ATP is released after the conformation changes to the O, that is open. This is loose conformation, tight conformation and open conformation. During this open conformation, ATP molecules are released. So loose to tight and then tight to open. Again this open molecule is again enlarged to become loose, wherein ADP and phosphate come and bind. The cycle goes on continuing until the protons are being effluxed from the inner membrane space to the matrix. So this ATP synthase enzyme is known as world's smallest molecular motor. It is smallest molecular motor that is present in the biological systems. So now coming to the inhibitors of oxidative phosphorylation. As we have learned, inhibitors of ETC, those are known as uncouplers. These inhibitors of oxidative phosphorylations are also known as uncouplers. So what are the characteristics of these uncouplers? So these uncouplers, they increase the permeability of mitochondrial membrane to the protons. As we know that the mitochondrial membrane is highly impermeable to proton. So these uncouplers, what they do, they increase the permeability and they allow the protons to come back again into the matrix. There is no need to travel from the ATP synthase. Directly they can come from intermembrane space into the matrix because the permeability of mitochondrial membrane is increased. Second is, whatever the energy that is being generated during the transfer of electrons from one complex to the other during ETC, it is dissipated as heat. There is no capture of energy for transfer of protons, and that whatever the energy is being synthesized, it is directly liberated as heat. There is no capture and no transfer of protons. And the third characteristic is they allow the oxidation of substrate, but there is no formation of ATP. We know that NADH and FADH2 undergo ETC and undergo oxidation. So this oxidative mechanism is facilitated but without ATP formation in presence of uncouplers. So these are the characteristics of uncouplers, that they increase the permeability, they release the energy in the form of heat, and they allow oxidation of substrate without ATP formation. So what are the examples of such uncouplers? One of the example is 2,4-dinitrophenol. The other is dinitrocresol, pentachlorophenol, and also trifluorocarbonyl cyanide phenyl hydrazone. So these all are the external agents that acts as uncouplers. But also body contains some internal membranes, or internal components, or you can say physiological compounds that act as natural uncouplers. So example of such physiological uncouplers, or also you can say natural uncouplers, is thermogenin, thyroxine and long chain free fatty acids. So this thyroxine is a hormone that is released by thyroid glands. Long chain free fatty acids are obtained after degradation of cholesterol in the body. And this thermogenin, it is a component of brown adipose tissue. This brown adipose tissue has a significant biological role in the body. So this brown adipose tissue concentration is high in case of lean people. The people who are very lean, even though after having a proper diet and after having a proper food, their weight doesn't increase or they don't become obese. The reason is high amount of brown adipose tissue present in them. So this brown adipose tissue converts excess of food into heat. It doesn't let it convert it into fat. It doesn't convert any of the food consumed into the fat. Directly it converts the food, and the excess of calories or excess of food particles and whatever the metabolites are produced, they are directly oxidized and only heat is generated. There is no deposition of fat in the body because of presence of thermogenin, that is a part of brown adipose tissue. So what is the significance of this uncoupling? So significance is, brown adipose tissue, we already learned that these are specialized tissue that are rich in electron carriers and are specialized to carry out oxidation uncoupled form of phosphorylation. So now this presence of active brown tissue in certain individuals is believed to protect them from becoming obese, because the excess of calorie whatever they consume is burnt and is liberated as a heat instead of being stored as a fat. So this is the reason why the lean people, even after consuming good amount of diet, they don't become obese. Ionophores. These ionophores are lipophilic substances that promote the transport of ions across the biological membranes. So ionophores, ions and phores. Phores means these act as a carriers, you can say, or these increase the permeability of the biological membranes, or they may act as a pores or phores. So iono means they transfer only the ions. These are the lipophilic substances that transport ions across the membranes by acting as a channels also, you can say, or acting as a pores in the biological membrane. So all the uncouplers, whatever we have listed, that is ETC as well as oxidative uncouplers, all are known as ionophores because they transfer the ions and increase the permeability of a membrane so that ions are being transferred easily from one space to the other space. So this is regarding oxidative phosphorylation, how ATP is being synthesized by using rotary model of a ATP synthase that was proposed by Paul Boyer. Hope you have understood. Thank you.